Hi everyone, welcome to Dento'ZEN. Today we are here with our second video on microbiology where we are going to discuss the morphology and physiology of bacteria. Morphology refers to the appearance, that is size and shape of bacteria. Physiology refers to the various cell processes inside the bacteria, like how do they grow, how do they multiply. This topic is further divided first into classification, second different types of microscopes by which we can see bacteria. These can be short notes for your exam. Third, different staining methods by which we can stain the bacteria and see them under the microscope. Again, there can be different short notes on different staining methods. Different shapes of bacteria which are seen under the microscope, bacterial anatomy, the structure of bacteria, cell wall, organelles, flagella. Again, there can be short notes. And then finally, the growth and multiplication of bacteria, where we will discuss the bacterial growth curve, which can be short note for your exam. So in today's video, we'll discuss the classification and types of microscopes. Remaining topics we'll take in our next videos. So let's start. Before starting, quickly subscribe to Dento'ZEN, tap on the like button and hit the bell icon so that you remain notified about new videos. First, the classification. Microorganisms belong to a heterogeneous group of different living organisms. First, they were classified into plant and animal kingdoms, but this classification was not satisfactory, so they were further classified into third kingdom, which is known as Protista. So remember, microorganisms belong to the kingdom Protista. Now, Protista can further be of two types based on the cellular organization and biochemistry. They can be prokaryotes or eukaryotes. Prokaryotes are simple organisms, eukaryotes are complex organisms. Examples of prokaryotes can be bacteria and blue-green algae, both starting with B, and for eukaryotes it can be fungi, other algae, slime molds, and protozoa. These examples can be important for your entrance exam questions. Now, bacteria lack chlorophyll and they do not show true branching, but there are exceptions, and the exception is actinomycetales. Now, talking about the differences between prokaryotes and eukaryotes, which can be an important short note for you. First, about the structure. As we look at the diagram, prokaryotes are simple in structure, they are unicellular, single cell, whereas eukaryotes, they are complex in structure and they are usually multicellular. Talking about the nucleus, proper nucleus is absent in prokaryotes, whereas it is present in the eukaryotes. And in the nucleus, we have nuclear membrane, nucleolus, we have deoxyribonucleoprotein, chromosomes, and mitosis. All these things are seen in eukaryotes, whereas they are absent in prokaryotes, where the nuclear material is known as nucleoid, which is a circular material. Now, if we talk about the cytoplasm of the eukaryotes contains so many organelles, as you can see in the picture. So there are endoplasmic reticulum, Golgi apparatus, then we have mitochondria, lysosome. So all these organelles are present in eukaryotes and they are absent in prokaryotes. Chemically, prokaryotes do not have sterols, so they are absent, but their sterols are present in eukaryotes, and muramic acid is present in prokaryotes, which we are going to discuss in further videos, and it is absent in eukaryotes. Then the examples we already know: bacteria and blue-green algae for prokaryotes, and for eukaryotes, fungi, other algae, slime molds, and protozoa. So for your short note on differences between prokaryotes and eukaryotes, you can use these differences and this diagram. Now let's talk about the size. Unit of measurement of bacterial size is micrometer, which is also known as micron. So 1 micron or 1 micrometer is 1/1000th of millimeter. So that is how we measure the bacteria. 1 millimeter we already know is 1/1000 of meter. Now, if bacteria are too small, we can use another unit, that is 1 nanometer, which is 1/1000th of micrometer. And we can use another unit, 1 angstrom, which is 1/10th of nanometer. Now, the resolution power of our eye, that is directly we can see a size of about 200 microns, but bacteria are too small. So let's see the size of bacteria: 0.2 to 1.5 micrometers in diameter and 3 to 5 micrometer in length. That can be Viva question. Compared to viruses are 20 to 300 nanometer diameter. Parasites, protozoans are micrometers, helminths are few millimeter to meter, and fungi, which have hyphae and thread-like structures, they are about 2 to 10 micrometers in diameter and several centimeters in length. So bacteria are much smaller than what we can see with our unaided eye. So they are seen with the light microscope, and viruses, which are further smaller, can be seen with the electron microscope. So let's now talk about the microscopy. Now, first let's talk about the important properties of microscope. First important properties, we want to see small bacteria, so we want to see them as an enlarged or magnified image. So important property is the magnification. So microscope should have good magnification, which is achieved by the use of lenses. Two types of lenses: objective lens and the ocular lens helps in increasing the magnification. When we look at a normal microscope, it has a base, it has a handle, it has a light source from which light is emitted, then condenser takes the light, it passes on to the specimen which is placed on this stage, and the specimen, the light is collected by the objective lens, and then this light goes through the ocular lens, and from there we see a magnified image in the eye. So that is the basic parts and the functioning of the microscope. So two types of lenses, objective lens and ocular lens, magnifies this specimen size and magnified image is seen at the eye. So let's see, ocular lens magnification power is 10x, and for objective lens we can have different types of objective lenses with different magnification powers. For example, we can have scanning which have 4x magnification, low power objective which has 10x, high power objective has 40x, and oil immersion objective has 100x magnification. All these can be your Viva questions. So total magnification is magnification of objective lens into ocular lens. So for objective lens, maximum that can be achieved is 100, and for ocular lens is 10. So total magnification can be 1000x. Now another important property is resolution. Microscope should have good resolution. That means if two cells are there in a sample, so and they are at a distance, so we should see them as two separate objects and not as a blurred image as one object. So you might have seen when you see a picture in your camera and you magnify it, sometimes it becomes blurred. So we do not want it to get blurred, and we should be able to see it as a clear picture. So that is the resolution. So it is the ability to produce separate images of closely placed objects, which is distinguished as two separate entities. Now light rays, so light source emits light which has wavelengths. So more is the wavelength of light, less will be the resolution. So we want our wavelength should be less so that resolution increases. So decreased wavelength in electron microscope increases the resolution in the electron microscope as compared to light microscope. Now these light rays passes through the specimen, and in the specimen if we are using the different mediums, for example air and oil, so based on the medium, based on the refractive index of the medium, resolution can be changed. So medium with higher refractive index, for example oil, can produce more resolution, can enhance the resolution power. So let's see at the resolution powers or resolution by unaided eye. With a direct eye, it is 200 micrometer size that we can see. With light microscope, it increases to 0.2 micrometers, and with electron microscope, it further increases. That is, we can see a size as small as 0.1 nanometer. So those can be your Viva questions. What are the different resolutions? Now, third important property of microscope that should be there is good contrast. That means if we are seeing a specimen, it should be well contrasted from its background. How do we achieve that? If the specimen is seen such, the contrast will be less, but if it is stained, then a good contrast is produced. So it is improved by staining. When stains bind to the cells, so contrast is increased. So we have learned three important properties of microscope. This first is good magnification, which is achieved by the use of lenses, objective and ocular lenses. Then good resolution by changing the refractive index of the medium. And third is good contrast, which can be increased by the staining. Now let's talk about the different types of microscopes. Bright field or light microscope, dark field or dark ground microscope, it can be important short mode. Then phase contrast microscope, fluorescent microscope, and finally the electron microscope. So let's discuss them one by one. First is bright field. As the word says, bright field. So the field, that is background, will be bright, and within which image that we are seeing of the object or the specimen will be dark. So remember, dark image is seen against a brighter background. So remember, brighter field, brighter background is here. Now let's see the working principle, how it operates. So we have a light source. From light source, the rays of light pass through the iris and the condenser, and they fall into the specimen. And from the specimen, the light rays are further gathered by the objective, collected by the objective, and passes through ocular lens, and a magnified image is seen in the eye. So here we can see image which is dark and the background is bright, or we can say it is light or white, bright background. So bacteria can be seen in the living state, that is, they are not stained, or we can fix them and stain them, or they can be stained. As we can see here, they are stained. So when they are seen in the living state, that is known as wet films or hanging drop preparations, where we just put the specimen directly into the drop under the microscope without staining, so we can see the living bacteria, we can study their shape, arrangement, their movement, their size, or we can stain them and can see them against the background. Now, important drawback of bright field is lack of contrast. Because the image is dark, we cannot see the details because the contrast with the surrounding background is less. So how do we increase the contrast? What we can do? If we somehow change the background and make it dark, so we can see this bacteria against this dark background, and we even do not need to stain it. So this self-luminous light bacteria will be seen against the dark background, and that is what is the principle of dark field or dark ground microscope. Now that can be important short note. So remember, dark field or dark background. So here the background is dark and the object is light. So object is light or bright against the dark background. Remember, the dark background, dark field. So how that is achieved? Let's see the working principle. That is achieved by changing the condenser into a special type of condenser which is called dark field condenser. So this condenser has a central circular stop inside it. So this stop, central opaque area, will not allow it will block the light from the light source. So these light rays are blocked, they do not pass through. But peripheral area of this condenser, it allows the light to pass through and focus on the specimen obliquely like this. So from this specimen, the rays which are reflected, they are collected by the objective lens. Here purple rays, they are collected by the objective lens, and unreflected rays are not allowed to pass through the objective. So unreflected rays here they are shown by black, so they are not passing through the objective. So only the rays which are passing through the specimen are collected, and thus the specimen becomes bright, luminous, and the background becomes dark. So that is how this is done. So specimen is bright, as you can see here, and background is dark. So background is dark. So reflected light is used instead of transmitted light in the dark field microscope. Remember, only the light which is reflected from the specimen is used. So applications of this type of microscopy are for identification of living cells, unstained cells. So we do not need to stain cells because they are self-luminous. For example, we can also see the structures like flagella can be seen, or very thin bacteria like spirochetes can be seen by the dark field microscopy. Now that can be your important viva question. Now next is the phase contrast microscope. The contrast between the specimen and the background can further be increased by a special type of microscope. So contrast is enhanced there because of the difference in the contrast between the cells and the water. So let's see how that is done. Let's see the working principle. So again, the condenser is similar to the dark field condenser. That is, there is central stop, central opaque area, central circular area which do not allow the light to pass through, whereas from the peripheral ring area, the cone of light passes through the cell, as we can see here, cell in the specimen, and from here some rays are bent and retarded by 1/4 of wavelength. So some rays are reflected rays, as we can see here, and some rays are unreflected rays. So two different types of rays are there which are passing through the objective. And then we have another important component, face plate, which is inside the objective. So from the face plate, these change in the wavelength of the rays which are passing. So we have two types of light rays, reflected light rays and unreflected light rays, and we can have two different types of waves of light which are different in their phases. So if we see here, so this purple is the reflected one, it is in different phase, whereas the black one, which is unreflected one, is in totally in different phase. So because of the change in the phase, there will be change in their appearance and change in the intensity which is perceived by the eye, and a contrast is created. So if we see here, we can see that there is this is in different phase, this is in different phase. So the two rays are in different phases, because of which a contrast is produced. So background will be bright and unstained object will appear dark against a bright background, so because of which a contrast is created. So applications of this type of microscopy is for seeing motility of bacteria. Living bacteria can be seen, which can be seen as moving bacteria, and then determining the shape of these living cells or bacteria, and also to see the internal structures within these cells as the contrast is enhanced, so we can see the internal cellular components of these microbes. So those are the applications of phase contrast. Next is fluorescent microscope. Here the fluorescent dyes are used, which are placed along with the specimen. So these fluorescent dyes, they have an important property. So whenever they take the light of particular wavelength, so they get excited. So they will take wavelength of short wavelength light rays, they will get excited, and finally they will emit the longer wavelength light rays, which will be of totally different color. So these longer wavelength light rays of different color are seen as a different color specimen in the eyepiece. So let's see the working principle. So here we usually use the mercury lamps. So these mercury lamps emits rays which pass through a special type of filter. So excitation filter is one important component. What is the role of this filter? So it will not allow all the wavelengths of the visible spectrum to pass through, but only short wavelength rays to pass through. So here we can see short wavelength blue rays are passing through this excitation filter. So that is the role of excitation filter. These excitation filter, the rays which are coming here, now they are going to the second important component, that is called dichromatic mirror. So from here, these rays, this dichromatic mirror, it allows the rays to pass into the objective and onto the specimen. So these rays, short wavelength blue rays, are passed on to the specimen which contains the fluorescent dyes. Now the fluorescent material, dye material, will now get excited and will emit light of another wavelength which has higher wavelength, for example green. So this longer wavelength light rays are emitted which passes through objective, and this dichromatic mirror now allows these light rays to pass through them. So excitation filter allows only short rays to pass through them, and then these exciting rays are falling into dichromatic mirror, fall on the specimen with fluorescent dye. From there, the higher wavelength light rays are passing through the dichromatic mirror into the barrier filter. That is the third component. Now what is the role of barrier filter? It allows only the longer wavelength light rays to pass through, so that we can see only this wavelength light as green colored specimen, and it will remove other blue or UV light. So that is how the fluorescent microscope works. Now let's see the application. Certain organisms which autofluoresces, which can show this property, that is they can take the shorter wavelength light rays and can of different color and emit the longer wavelength lights of different color. For example cyclospora. Then we can put the fluorescent dye in the microbes and we can see them in different colors. And then the phenomena of immunofluorescence, where we can label certain proteins within the organisms or the cells, and we can see those proteins, immunocomponents, with the help of these fluorescent dyes. So those are the applications of fluorescent microscope. Finally, we come to the electron microscope. As the name says, here we are using electrons. So it was invented by German physicist Ernst Ruska in 1931. Source of illumination is electrons as compared to the light in the light microscope. So that is important difference that you have to remember. So we also said that shorter is the wavelength of light, more will be the resolution. So here the electrons which are used, their wavelength is shorter, this much shorter than the visible light photons which are used in light microscope, because of which it has far better resolution than the light microscope. So we can say that electron microscope has more resolution, better resolution. Let's see the working principle. So electrons are the source, so we need electron gun which will produce the electrons. So these electrons are produced and these electrons have to be projected onto the specimens. Now here the type of lenses which are used, which are different. For example, here we are using electromagnetic lenses as compared to the lenses made up of glass in the light microscope. So here we are using electromagnetic lenses. So these lenses will project these electron beams onto the specimen. So they will focus these electrons from the specimen. These are all collected by the objective, and from there an image is collected and is formed on the film, photographic film. So here the medium which is used is vacuum. That is, for example, in light microscope we were using air, but here we are using vacuum because electrons, if they will pass through air, they will collide with the air and they will change their direction, and we do not want them. So we use a vacuum medium. So we can only see dead and dried objects here in the electron microscope. Now two types of electron microscope are there, transmission and scanning. So transmission electron microscope, the electrons passes through the specimen and they are then projected into different directions, and whereas in scanning electron microscope, as the name says, it scans. So it just scans the surface of the specimen. For example, from the different areas of the surface, different types of rays will be emitted and that will produce a contrast. So we can study the surface of the specimen using the scanning electron microscope. So we can say the applications of electron microscope for virus detection, and because they are very small, so the resolution power increases with electron microscope, so we can see small viruses. And then with the scanning electron microscope, we can study the surface of microorganisms. Now if we talk about the differences between light and electron microscope, which can be your viva question and as well as short note. So that first, the light microscope, the source of illumination is light, whereas in electron the source of illumination is the beam of electrons. Then talking about the type of lenses which are used, condenser, objective, ocular, they are made up of material which is glass, whereas in electron microscope they are made up of electromagnets. Then talking about the medium, air is used as medium in light microscope, whereas in electron microscope a vacuum is used. Then specimen is mounted on a glass slide in case of light microscope. For electron microscope we use a special type of metal grid, mostly made up of copper. Then highest practical magnification that can be achieved with light microscope about 1000, whereas with electron microscope it is much more, and the resolution is more with electron microscope, that is 0.1 nanometer as compared to 0.2 micrometer in light microscope. Now let's check what have you learned. So electrons are used as source of illumination in which microscope? Light, dark field, electron or phase contrast microscope. Second question, in dark field microscope, object is dark, background is dark, both are dark or none of the above. So that is all for this video. If you really enjoyed the video, do tap on the like button and share this video with your friends. Keep watching, keep learning, keep smiling. See you in the next video on microbiology soon. Till then, take care. Bye bye.